making sense. So firstly, we've got our classical pathway, and from that I'm going to try and talk about the alternative pathway as well as lectin pathway. So they're three different pathways, however the end like final few steps are all the same, and the main difference is where they all start from. So the classical pathway, I think that's straight. The classical pathway, it starts from our antigen and antibody, the reaction between both of those. And what this does is this activates C1. Now, also, by the way, all the C's, they stand for like complementary, so it's just like a reference. So C1 then will go into C4. Now C4 then splits into C4A, C4B. Now C1 then combines with C4B, combines with C4B, and now this will form our C14, I think, or maybe that's too small. Let me just make that a bit bigger. C14B. C14B, this is an enzyme, and that will allow for C2 to split into C2A and C2B. Now C14B and C2A form C14B, C2A. This is an enzyme, aka C3 convertase. Now this is going to be a massive reference point in terms of the other two pathways. Now C3 convertase, this would then allow for C3 to form into or split into C3A, C3B. Now C3B, this is an important part. C3B then would form along with our C3 convertase to form C14B 2A 3B. Now this is also known as C5 convertase. And now C5 convertase would allow for C5 to turn into or split into C5A, C5B. C5B is a bigger, and C5B then combines with C6, C7, C8, C9, and this will form C5A, 6, 7, 8, 9. And this is also known as our membrane attack complex, also known as MAC. And what this does is this will cause our cell lysis. So that's our main part. I wonder how long that part took. I'll find out later, I guess. Then we go into a different pathway. Let's go into alternative first. So I made a few notes prior, so because this takes a bit of time. So alternative pathways, this is triggered mainly by bacterial endotoxin. So this has a lipopolysaccharide, and that is found in gram-negative bacterial cell walls. As well, that's the main trigger of the alternative pathway, as well as yeast cell walls, and I don't know what this means, but cobra venom factor. Allahu alam if I find that out what that is later. So basically, alternative pathway, it starts with the spontaneous hydrolysis of C3 factor C3, and now this is mostly found in your blood plasma. Factor C3 is then converted, here, into C3b. Now factor C3b is then combines with factor B, and for whatever reason that then splits, and to allow the splitting we've got our factor D. So factor D allows for factor B to be split into BA and BB. Now our C3b and our BB will then form C3BBB, also known as our C3 convertase. Yeah, that's more or less the same part. So C3 convertase, and then this will then go forward to C5 convertase, and then finally into our membrane attack complex. Now, the structure is a tiny bit different from this one. However, this would also be C3 convertase, and the role is the same, and the pathway after that would then be the same as well. Now, lectin pathway, this is a bit more complicated, and there's a few like bigger words. So a few key words we've got are MBL, also known as our mannose binding lectin, as well as MASP, which is our mannose binding lectin associated serine protease. That seems like a lot of waffle. I guess I'll find out later a bit more about that. So basically what happens is we have these type of pathogens. I don't know how to pronounce these, Neisseria, Candida, and Salmonella. So these are all pathogens. These have got glucose as well as mannose in their cell wall. So the body would, so if these pathogens enter the body would detect these, thus it would release, I think MBL is produced actually in the liver. Somewhere have I got, okay, yeah, anyway. So basically what would happen is then the MBL, which is released, I guess, would then combine with MASP, which is a mannose binding lectin associated serine protease, and then this would form C4 and C2. These would then split into C4A, C4B, C2A, C2B. Now these two, C4B and C2A, they would combine to form C4B, C2A, and you can probably guess it. This is also this enzyme would be known as C3 convertase. Now this would then follow the same path to then form C5 convertase. When I say same path, I mean like, so C3 convertase then breaks down C3 into C3A, C3B. That then C3B then combines back with the original C3 convertase. Then that would form your C5, and then that gets split to thus form your membrane attack complex. So that's all of that in terms of that. And then a few important things from this whole topic, I think, which I think was on one of the slides, is a few important points. So C3A and C5A, these are our chemo attractants. So that is, if I get a little green pen, so C3A is here. Actually, let me get pink instead. So I'll just cover over that. So C3A, and then we've got our C5A. These would be our chemoattractants. Then we've got Then we've got C3B. This would be involved with opsonization, which I believe is linked closely with phagocytosis, and then it releases something on the cell surface. I'll find out more about that later. Then we've got C3B. C3B is here. And then we've got finally C5B. And the role of that would be kickstarting the MAC, or membrane attack complex. And C5B is here, of course, that then causes the attack of the